Christmas will never be the same after the family gathering in 2008. Instead of a cheerful time, it was full of fears and prayers. Cancer was the word of the night. Acute myeloid leukemia is a fast-growing cancer originating in the developing cells that would normally be classified as white and red blood cells. In many AML cases, a mutation to the gene c-KIT can cause uncontrollable growth of harmful cells, flooding the body with cancer cells. c-KIT mutation occurs in 20 to 25% of AML cases. My father was a part of that 20%.
c-KIT is a receptor protein tyrosine kinase. It's just a normal gene that when mutated becomes cancerous. When binding its ligand, which is a substance that forms a complex with a biomolecule, it forms an active dimer, which is a molecule or molecular complex that links two identical molecules. Transphosphorylation occurs. When something transphosphorylates, a phosphate group is transferred from one molecule to another. The addition of the phosphate group comes from the nucleoside triphosphates such as ATP. When the transphosphorylation is complete, proteins remove the phosphate groups, activating uncontrollable cell growth that causes the growth of a tumor.
In AML specifically, the mutation of c-KIT occurs on exon 17.
According to an article written by Johan Lennartson and Lars Ronstad on stem cell factor receptors, the active and inactive state of c-KIT kinase domain has been found within the molecule. Overall, the kinase domain of c-KIT conforms to the general kinase fold with a primary amine terminal lobe and a carboxylic acid terminal lobe, with the active site located in their interface. p13 kinase is activated by stem cell factor both through direct binding to tyrosine 721 in c-KIT and indirectly through binding to the tyrosine phosphorylated adapter protein GAB2.
In the same journal, Stem Cell Factor Receptor/c-KIT, From Basic Science to Clinical Implications, written by Juan Lennartson and Lars Ronstad, these findings were found. In summary, c-KIT binds to a kinase structure with two lobes, with the ATP binding and active site occurring in the interface. In the inactive site, the juxtamembrane region forms a hairpin loop that inserts into the active site and interferes with the C-helix and ATP binding P-loop. This slows kinase activity. c-KIT dimerization is driven by a ligand-induced process that is stabilized by interactions between immunoglobulin-like domains and proximal receptors.
c-KIT mutation analysis uses allele-specific polymerase chain reaction and sequencing for prognostic stratification of AML. The activating mutation D816V in c-KIT is associated with the worst prognosis. The test is taken from whole blood or bone marrow extracting 1 to 2 milliliters. c-KIT is considered a main target for molecular cancer therapy, but is currently being treated with chemotherapy and radiation.